Hello students, welcome to the channel. We have been working on the reading section of the April 2021 SAT in the last couple of videos. Today we'll discuss the third passage pertaining to questions 21 to 31. This passage is adapted from Chris Brody, No Use Moving the Cheese by Sigma C, the Scientific Research Society. Superman has super healing. Spider-Man has an uncanny spider sense. But truth can be stranger than fiction. The newest superhero doesn't wear a cape or mask. It's a mouse, and it looks just like its normal brethren. Its superpower is its amazing nose. In a paper published in Neuron, collaborators at Florida State University and Yale University describe what they call super smeller mice. These exceptional creatures have noses that are 1,000 to 10,000 times more sensitive than those of ordinary mice. So the first paragraph introduces this idea of super smeller mice, which have been studied at Florida State University. And these creatures have a smelling sense that is 10,000 up to 10,000 times more sensitive than that of ordinary mice. I should also mention here that the title comes from this famous book called Who Moved My Cheese, which is basically a business book about unexpected changes in business. But the reference in the title is to that book, right? The superhero origin of these rodents involves the deletion or knockout of a gene. This technique usually generates mice that are quite sick, as nearly all mutations are harmful. Yet it doesn't seem to be true for this gene, KV1.3, which encodes a protein that acts as a channel to let potassium ions into cells. This particular ion channel is found in immunological T cells and neurons in the hippocampus and the olfactory bulb, the part of the brain that gets information from odor receptors in the nose. So this kind of starts to explain the biological basis for why these super smeller mice have such a strong sense of smell. It's because of this genetic mutation, right? All mutations are harmful, but not in the case of this genetic mutation in the KV1.3 gene, which encodes a protein that acts as a channel, allowing potassium ions into cells, right? In neurons, K plus channels, such as K1.3, can act like governors on an engine, restricting the firing rate of the electrical spikes known as action potentials. The deletion of KV1.3 removes this block using mice generated in the Yale lab of Richard Flavel. A team at Florida State led by Deborah Fadul discovered that the loss of the channel caused one type of olfactory neuron, the mitral cell, to fire at lower thresholds and higher frequencies. Furthermore, the mutant cells were insensitive to chemical messages that normally rein in the flow of electrical current during an action potential. According to co-author Leonard Kaczmarek, whose group at Yale studies the ion channel biology of sensation, these changes resulted in greater excitability and better timing, effectively phase locking the output of the mitral cells and thereby increasing the coherence of the olfactory signals. So this paragraph describes the process of how the blocking of K1.3, the deletion of K1.3 removes this block that permits a sort of rein, the block that permits a sort of rein on the intensity of the electrical signals that are fired in the brain. And because that block is removed, you can see increasing coherence of olfactory signals, smell receptors, and that is why the mice that we are talking about have such strong sense of smell. The mutation also caused structural changes in the olfactory bulb. In this part of the brain, olfactory receptor cells connect to mitral cells in clusters called glomeruli. The knockout mice had glomeruli that were about half as large but twice as abundant as normal. As a result, information from the nose went to twice as many processing units as usual. Fadul suggests this might increase the resolution of the signal, meaning that a faint odor would be more likely to be noticed above the jumble of background smells. So this paragraph explains how this mutation also caused structural changes in the olfactory bulb, the part of the brain that deals with the sense of smell. The knockout mice had glomeruli that were about half as large but twice as abundant as normal. And this meant that information went to twice as many processing units. And so even faint smells that would otherwise be ignored could be picked up by these mice. Mutant mice could distinguish between complex odors such as peppermint and powdered food with nearly 15 times the sensitivity of normal mice. They were also better at detecting subtle molecular differences between odorants, such as some, but not all closely related alcohols. The most amazing change was a huge increase in sensitivity. Mutant mice were able to perceive an odor that was 1,000 times more dilute than what wild type mice could smell. The super smeller was definitely a surprise. None of the investigators intended to create such a creature. We had no inkling, states Kaczmarek. We were looking for an effect in the auditory system. So they were looking at something to do with the ear, the auditory system, but this turned out to be a surprise. Okay, we'll look at the chart later, right? We'll just get to the questions first. The primary purpose of the passage is to describe the discovery of a new species of mice, explore the results of a genetic mutation in mice, consider the roles of different genes in mice, discuss a misconception about the sense of smell in mice. So I think the best answer is B. You are looking at a genetic mutation which has resulted in a strong super smell ability in these mutant mice. So that's the mutation that we are discussing. It is not a new species of mice, so A is wrong. We are not looking at the role of different genes. We're only looking at this one mutation. And we are not discussing misconceptions about the sense of smell. So B is the best answer. In the first paragraph, the references to Superman and Spider-Man primarily serve to. Now, this is a very common question on science passages. Most of these science passages are written for lay readers, and they are meant for the lay reader to kind of understand this scientific research or scientific experiment that's described and to make it relatable to the reader, right? So you would always have expressions or phrases that inject a sense of humor, a sense of relatability. So to emphasize the unusual nature of a research finding, explore the practical implications of a recent discovery, define the use of a scientific term by providing familiar examples, provide a contrast between realistic and imaginary situations. So it's C, right? Because you're trying to describe what this super smeller mutation is going to be. And you're kind of trying to say that it is similar to what Superman and Spider-Man, the kind of the abilities that they possess, right? So it's only to kind of introduce the idea of this mutation by using a familiar example. It's not the unusual nature. It's not the practical implications. Superman and Spider-Man are imaginary creatures, so it's not practical. And it's not contrast between realistic and imaginary. A, B, and D kind of give a lot more weight to those examples than the passage intends to. So it's just to, use familiar examples to introduce an idea. Okay, which choice best supports the idea that super smeller mice can perceive distinction in odors that are very similar to one another? So I think this was here somewhere, right? The most amazing, one second. The most amazing change was a huge increase in sensitivity. Mutant mice were able to perceive an odor that was 1,000 times more dilute than what wild type mice could smell, right? So that is, they can oh, this is not similar. Okay, no, not this. Is it this? Mutant mice could distinguish between complex odors such as peppermint and powdered food with nearly 15 times the sensitivity of normal mice. They were also better at detecting subtle molecular differences between odorants, such as, okay, so this is, so it's definitely not A, because that's not the part where we talk about 19 to 23, right? Yeah, this is not, this is describing the scientific basis of what was found. 35 to, he increased the resolution. Yeah, it's not about, this line's option B is not about similar odors. Mutant mice could distinguish between complex odors, okay, and 40 to 42, yeah. So it is D, because they are very similar, right? Closely related alcohols. Closely related alcohols would indicate that those are very similar, and yet super smeller mice could distinguish between them. As used in line 20, generated most nearly means the deletion of KV 1.3 removes this block using mice generated in the Yale lab. So they were kind of produced, right? They had the mutation, so produced, right? That's, the mice were produced in the lab with the mutation, right? So it's not provoked or performed. It's not energized. The mice are produced. So it's C. As used in line 23, insensitive to means, furthermore, the mutant cells were insensitive to chemical messages that normally rein in the flow of electric current during an action potential. They were insensitive to chemical messages. That is, they did not respond to chemical messages. They were unresponsive to, right? So the channels that kind of rein in the flow of electricity, these mutant cells did not respond to those channels, and that is why the higher coherence of olfactory signals. It's not unconcerned, it's not unemotional. We are not talking about emotional or such responses, and it's not merciless. So it's C. Which conclusion about the functioning of the sense of smell in mice is best supported by the passage? The fewer the number of mutant cells, the more powerful the sense of smell is in mice. The greater the number of olfactory receptor cells connected to mitral cells, the less sensitive the sense of smell is in mice. The fewer the number of glomeruli, the less sensitive the sense of smell. The larger the size of glomeruli, the more powerful the sense of smell. So I think it's C, because here you have the knockout mice had glomeruli that were about half as large, but twice as abundant as normal. As a result, information from the nose went to twice as many processing units, right? So if the number of glomeruli is fewer, then the sense is also less sensitive. And this comes from line 32 to 35, right? So 32 to 35, that's option D, where we explain that, you know, the more processing units that they can access depends on the number of glomeruli. According to the passage, the discovery of the super smeller mice was unexpected in part because the researchers did not know that the KV1.3 gene existed before they pursued the experiment, were originally investigating the relationship between KV1.3 gene and the mice's hearing. And that's correct, right? None of the investigators intended to create such a creature. We were looking for an effect in the auditory system, right? They had intended to show that KV1.3 gene did not control the sense of smell and believed that normal mice possessed a more acute sense of smell than they actually had. So it's very clearly B, because they were interested in the role of this gene on hearing, but they found that it actually affected smell. According to the figure, the mean percent of the wild type mice at retrieving the food reward was no greater than 50% for which of the following peppermint odorant dilution factors. Okay, so this is the mean percent success of mice at retrieving food reward buried in litter, scented with varying dilutions of peppermint odorant. And we are looking at wild type mice, which is black, and the KV1.3 deleted mice, which is gray. So for the wild type mice, it was no greater than 50%. which means that it would be, it would have to be these last three, right? Because for others, it is always present. For these last three, it was, for which of the following, it would be 10 billion and higher, right? So 10 billion and higher. So D is the best answer. If when it is less than that, they could kind of distinguish the smell. Okay. According to the figure, at which peppermint odorant dilution factor did the mean percent success at retrieving the food reward remain above 90% for both KV 1.3 and wild type? So above 90% is for 100, right? Because if you have 100, then it's above 90% for this, for both, right? So this, at this dilution factor, both mice performed at the same level. According to the figure, which statement best supports what the author considers the most surprising, the most surprising finding about the mutant mice perception of odor compared to that of the wild type mice as represented in the passage? Okay, let me read this. Both wild type mice and KV 1.3 deleted mice had an increase in success in retrieving the food reward when the peppermint odorant dilution factor changed from 10,000 to 100,000, right? They both had an increase. So that is kind of, yeah, surprising, but I don't think the author is concerned with wild type mice. The wild type mice had an increase in success retrieving the food reward when the peppermint odorant factor was changed from 1 million to 10 million. Whereas the KV 1.2 deleted mice suffered a decrease in success when the change was made. Okay, that's not something he's interested in either. The wild type mice had better than 50% success in retrieving the food reward up to a peppermint dilution factor of 1 billion. Whereas KV1 had better than 50% retrieving food reward when the peppermint odorant factor was greater than 1 billion. Okay. Wild type mice had the same success retrieving the food reward. Peppermint factor was diluted by a factor of 100. So I think it's C, because if you look at up to 1 billion, they did have a greater than 50%, was greater than 1 billion. So even at greater than 1 billion, it was higher. So they were interested, the researchers interested at the level of sensitivity. And that level of sensitivity is proved to be true even for dilution factor of greater than 1 billion for KV 1.3 deleted mice. So it's C. Okay, so that was the third passage. Let's grade this. We start from 21, right? Okay. And we go up till when? Till 31, is it? Yeah, so I'm just going to keep it till 31. Okay. BADC, BADC. Okay, so 22nd is supposed to be A. Okay. Okay, so I seem to have gotten this one wrong. We'll come back to this. Wait, I'll just write this. Okay. DC 25, CCDB, CCDB, 29, DAC. Okay. So in fact, I'm surprised that I got 22nd wrong, because I did say that this is a very common question type on the science passage. And the right answer is option A. It's the unusual nature of a research finding. And I think the reason for that is that they are trying to kind of compare it to these really, really extreme examples of Spider-Man and Superman. And you wouldn't normally associate mice with such, examples or such comparisons. And because these mice have turned out to be super smeller mice, I think the author felt the need to use these comparisons. So the idea of trying to make it relatable still exists, but the reason is that it's to emphasize the unusual nature. Why C may be wrong is because I think the scientific term about the mutation is not brought up in the first paragraph, right? So it's not that they are describing the scientific term by using the examples of Spider-Man and Superman, right? So, even though the approach that I was going into the question, which was the relatability, is correct, I think the particular answer choice C is not correct, right? And A is the best answer because the unusual super smeller capability is kind of supported with the reference to Spider-Man and Superman. Okay, so that was the third passage, science. Hope this exercise was useful. If it was, hit like and subscribe to the channel. I will link to the other solutions to the first two passages that have already been uploaded in the description box below. Do come back and check for the remaining solutions. I'll see you guys soon. Take care. Bye-bye.